I'd like to welcome everyone to CITRIS Research Exchange seminar series. And for those of you who may not know, this is the 20th year of CITRIS. And this one is the 13th year of CITRIS Research Exchange that we have hosted during this 13-year period, a remarkable lineup of technology innovators in person. And this year, because of the COVID-19, in the spring series, it's done, the exchange seminars are done virtually. So having said that, it gives me a great deal of pleasure and honor to introduce Professor Cristina Davis. Professor Davis is Warren and Lida Gitt Endowed Professor and Chair of Mechanical and Aerospace Engineering at UC Davis. She joined UC Davis after a time working at Draper's Lab in Massachusetts. So she does have she brought with herself some industrial experience to UC Davis. Her research is on designing sensors and instrumentations using MEMS technology to detect chemical biomarkers that are indicative of disease in human, animals, and plants. She heads up the BioMEMS and Bioinstrumentation Lab at UC Davis. She's also the Associate Director of UC Davis NIH-funded Center on Translational Medicine. She is serving as a member of the Scientific Advisory Board for United States Air Force. She is fellow of 3 societies, including National Academy of Inventors, and she has co-founded and is a scientific advisor to a UC Davis affiliated startup that was became into being due to her research. She's also the chair-elect of the International Association of Breath Research. It is with my great honor and pleasure to welcome Professor Cristina Davis, who is going to talk about using exhaled breath to diagnose COVID-19. Cristina.
Laura, thank you so much for the great introduction. I'm so delighted to be here today to talk with the people in the CITRIS community. Let me just get my slides up. Okay, so as mentioned in the introduction, I'm going to be talking today about using exhaled breath as a way to diagnose and to monitor for COVID-19. This is an emerging area and there's a lot of exciting activity on it. So I'll just launch in and give you guys an overview of the field, but also what I'm up to and what my group is up to. So we know that exhaled breath has a lot of health relevant information. When we exhale, we breathe out chemicals. Some things that we exhale are like carbon dioxide, things that we know about, oxygen, nitric oxide, respiratory gases that are traditionally measured. But we also exhale thousands of metabolites that could be very important in understanding our human health, but also what we've been exposed to in the environment. Now, when we exhale, we actually have multiple fractions of the breath. Some of them are gas phase and volatile compounds, and some of them are non-volatile compounds that are in aerosols. I'll talk about this fraction in just a minute. In terms of the advantages of breath analysis is that you have this way where you could potentially non-invasively monitor people because we're all exhaling all the time. And we could potentially use advanced sampling and measurement systems to be able to determine what's in breath to tell us information. So when we exhale, there are multiple fractions of what we exhale. We have those traditional respiratory gases, carbon dioxide, nitric oxide, and then also oxygen. We don't use up all the oxygen we breathe in, so we breathe a little bit of that back out. In addition to that, in the gas phase, there are these volatile organic compounds. There have been around 1,000 of those measured in the literature, not all at the same time. They tend to vary over time and they vary with specific clinical conditions. Now, in addition to the breath gases, there's also exhaled breath condensate. This is what you collect if you exhale and cool the breath at the same time where you're condensing it. So you get a liquid. It's kind of like when you exhale against a mirror or a piece of glass and you get a film that's on that glass or the mirror, that's condensate. It's a very small amount just from one breath. But if you do that in a specific way, and I'll talk about how we sample in a little bit, you're actually collecting a few different things. You're collecting lung aerosols. These are particles that are on the order of submicron. So they're anywhere from, submicron to 1 to 2 micron in diameter. And they contain a lot of content. So they're liquid-based as we exhale them, but they're coming from the liquid lining of the lung. And so they have everything in it almost that's in blood. It's just more dilute. And so those aerosol fractions are really interesting. They're not saliva, they're not spit. You can collect that from breath as well. It depends on if somebody is talking and you're collecting exhaled breath. But generally, we try to avoid saliva and spit because they have enzymes in the saliva, and that affects our ability to measure the different metabolites that are in it. So how you sample gets to what you see when you look at exhaled breath, when you examine it. There are a range of molecules. Some are very small and they're very volatile. Some are very large and they're low volatility. And so this is a combination of the sampling temperature and then also how you mechanically sample when you're collecting that breath fraction. And I'll talk about our protocols and how we approach it in just a minute. So when you look at exhaled breath condensate, it's really fascinating because we found between a little over 3,500 breath compounds to date. And this is both exogenous, endogenous compounds. So things that are generated in the body, but also stuff that's from outside the body, like drugs or other compounds. Those show up in the breath, and they show up specifically in exhaled breath condensate, because this is typically larger molecules that are dissolved in those little liquid droplets, the aerosols that we're breathing out all the time. Now, in addition to what's in exhaled breath condensate aerosol, you also see things like DNA, nucleic acids, and some pathogens, because pathogens not only inhabit, our respiratory tract, but they also are detectable there. It's one of the ways that those aerosol fraction droplets are one of the ways that pathogens can spread. A key example of this is tuberculosis. That's one of the primary ways that that particular bacterial infectious disease is spread. So we know that there's a lot inside of breath. And if we look at specific compounds, we are seeking to determine, very much like you would do with untargeted metabolomics of blood or urine or anything that comes out of the body, we're looking for biomarkers of disease. And so you could specify how you organize your clinical trial, and you could determine what diseases you might like to try to tackle. But at the end of the day, you're looking for fingerprints of chemicals that are associated with and that have a high probability of being associated with a specific clinical indicator. So for us, we're looking for some biomarkers that we know, but we're also open to finding biomarkers that we do not know about. Like right now, I don't know the identity of every biomarker for a specific infection, but later I could actually discover that when I look at the metabolites that are in the exhaled breath fraction. And this is really key because while COVID-19 and other infectious diseases are definitely detectable by some methods, in most of the methods we're using so far, we're looking at the specific virus. So when those methods are used to detect, they're detecting the virus. We're not detecting the virus. We're looking at metabolites that are indicating whether somebody has been infected. They're kind of the result of being infected. The virus is not part of what we're measuring. So once we know what chemicals we're looking for, the metabolites, then we can identify them using many types of sensors. Now, this could range. It could be a gas chromatograph mass spectrometer. It could be an ion mobility spectrometer. It could be a lot of different types of technology that could be appropriate, including there's a category called e-noses, but that's just one of many. So once we know the chemicals that are in that fingerprint of whether somebody has an infection or not, then we try to detect it. Almost always, this type of data ends up being multidimensional. It's very complicated. And we have found, and others have as well, that you almost always have to do some type of advanced signal processing or potentially using things borrowed from the field of artificial intelligence, pattern recognition, to be able to determine one type of signal versus what is a common background. So this is part of the key step for how we would use breath analysis in a clinical setting. Ultimately, the clinicians that are going to make use of this data, they need it to be distilled even further. They need a system that can actually take data that's very complicated like this and actually translate it into actionable data. So, you know, red if you're sick, yellow if you don't know and you need to retest, and green if you're healthy. So we actually have, these are the key steps that you would need to go through looking at breath analysis for COVID-19, among other types of infections. So we know that breath analysis as a concept has been around for quite a while. Originally, it was reported in both ancient Chinese medical literature as well as ancient Greek medical literature. There were reports where people associated specific smells on the breath, and that tended to correlate with a specific type of clinical condition. And so, you know, they were making the use of technology they had in their toolbox, which was their own sense of smell. We've come much further than that. A huge step was along the way in the 1700s, two different French scientists, Lavoisier and Laplace, they actually came up with a way to show the first chemicals in exhaled breath. They had this person sitting over here breathing into a mask. They routed their exhaled breath through this bubbled solution here, which happened to have bicarbonate in it, and then it came out the other end. What they found is as the carbon dioxide in the exhaled breath built up in that bubbled solution that the breath was going through, eventually the bicarbonate would precipitate, which is a basic chemical reaction. But believe it or not, that was the first time that we knew that you exhaled a chemical, and it wasn't just air. It wasn't just the stuff that we breathe in all the time and exhale. So this was a key fundamental step. We exhale chemicals, some of which are important. That one, carbon dioxide, is a respiratory gas, but now we know there's many others. You flash forward into the 1990s, and there have been, at this point, several reports of people sampling breath and also detecting chemicals. And at this point, our chemical library of what was in breath probably was on the order of magnitude of between 200 or 300 and 500 or 600 chemicals. But it took quite a bit of instrumentation to be able to take those measurements. This is from a scientist, Michael Phillips, who's a researcher on the East Coast. He's a friend of mine. And you know what he found early on was that you had to have some very specific sampling. The way that you did those gas measurements, the way you collected the breath had the potential to affect the measurement, so you had to standardize it. So he came up with a way to take some of these volatile organic compounds where they were doing what are called end tidal measurements, where you exhale and the last fraction of your tidal breathing is captured as a breath volatile snapshot. So it has whatever chemicals are in it, and it's the alveolar breath sample. So it's not the part in the trachea or in the bronchi, which are the large parts of our respiratory tracts, but it's the part that's down next to the alveolar, which is the gas exchange part, where the blood vessels are very rich, and that's where the major parts of our gas exchange happen from a physiological perspective. Some of the barriers that have been around for breath analysis have been around for a while. I always like to tell people, this is a slide I made back in 2002. It's 18, 19 years later. We still have some of the same problems, but we have made progress on each of these three areas. These chemicals that we measure in breath are really dilute. They're at very low concentration levels. And this is important to realize because it affects how we sample, but it also affects the types of technologies you have to use to measure them. So they are in what are called parts per billion concentration levels. They can be in parts per trillion concentration levels as well. Parts per billion, ppb, is about 1 spoonful of sugar in an Olympic-sized swimming pool. That's about 1 part per billion. If you were looking at parts per trillion, it's usually in the double digits, but then you would have this diluted by a factor of 100 or 1,000 more. So it's really dilute concentrations. That makes it very tricky to measure and to quantify. The other thing that was a factor for a long time is that we didn't know what we were looking for. We were kind of looking as a needle in a haystack. We had some things that we suspected were important, some biomarkers, but it was in this complex background that made it very hard to tell apart what you're looking for from kind of this confounding effect, because there's so much other stuff there. And so that needle in a haystack effect, we've actually made good progress on that as well, because you can actually now start to use advanced instrumentation to tease apart the chemicals that are showing up in the breath sample. We still have a lot of work to do, but it's made a lot of progress. And then finally, we needed for a long time device and sensor breakthroughs. This was lagging for a while because the people who first entered this field were basically doctors and chemists, wonderful research perspectives, but they didn't know how to design and manufacture specific instruments that are needed to do the types of research and to answer the questions that were in the field. Now, this is a very multidisciplinary area, and engineers have entered the area because I think they have a lot to offer in terms of the tools of how you can approach breath analysis. So one of the things that's been really interesting in the current pandemic is that early on, there was a recognition that some types of trained canine teams were able to detect COVID-19 patients in kind of public settings. And this has now been reported. I'm aware of at least 4 to 5 different teams. This is a few of them here, and there's probably more that I haven't heard of. And so an example of this is the Miami Heat, the sports team, is actually using trained dogs. These are not, these aren't people's pets. These are trained dogs like you see that screen at airports, except for in airports, they're looking for explosives and narcotics and other things like that. In this case, they're actually smelling odors to tell whether or not somebody may have been infected. They're not sniffing the virus. They're sniffing whether the reaction in someone's body from being infected changed how they smelled. And so one example of this is in the Miami Heat theater. When they let fans in, they actually screen them with dogs right now. And if the dog goes by and doesn't do anything special, then you get to go in and you get to watch the game. If the dog sits down beside you, then they have to take that person aside and say, hey, look, we're going to refund your money, but we're not going to let you in because the dog thinks you're sick. This is interesting, but it's a little bit unreliable potentially, because dogs, they get tired the same way people do. Their accuracies vary over time depending on what their tasks are. And so what was interesting about this is the recognition that there's a scent, there's a sniff, there's an odor associated with whether people are infected or not. That itself is really interesting. Chile, the country Chile, is doing some work in this area. Helsinki also doing this, and I read about a team in Germany. So this is happening all around the world, that people are training dogs using known cohorts. So they'll take patients that have known COVID-19 infections, and they'll take people that are not known to be infected, and they've shown that they are not infected. And so the dogs get trained, and then they get challenged with people where the infection status was unknown. What's interesting to me is that this is now showing up in the scientific literature, which I'm quite glad of, because anecdotally, this is great, but it's not science until you actually study it, in a very organized way. But there are some reports that are starting to show up in the peer-reviewed literature. That gives us a little bit more confidence in the types of analysis and the ways that people are doing this in their different settings. Interestingly, also, we'll kind of take a step back. Dogs can be great detectors. We know they have a much better sense of smell than us, but they do get tired. You have to have what are called dog handlers. So they're people who walk around with the dog, and they interpret the dog's actions. But this is a pretty people-intensive endeavor, and it's also can have problems, like if the dog gets tired, and it can only work so many hours a day. So looking at other types of analytical methods, kind of state-of-the-art chemical analyzers, is a very natural progression to move from doing canine odor detection to looking at how to do this in a much bigger scale, and in a way that can be deployed in many, many, many areas. There's a few areas, a few papers that have now come out. These all came out in the latter half of 2020. Obviously, because it took some time for people to get organized, but within a couple of months, you start to see groups in a few different areas that were starting to use state-of-the-art chemical analyzers. Again, this is in laboratory settings, but then you can worry about scaling it up a little bit later. There's a group that was actually in the UK. That's this group down here in the lower left-hand corner. Paul Thomas is the breath researcher that did this initial study with some scientists in Scotland and Germany. There's also a group out of France that started looking at this, and they also have a report of the odors that are being detected by the dogs. So they were trying to identify what were the things that the dogs were alerting on. And then also, of course, our group up at UC Davis is working in this area. And then this is a group that's in France, also working in the area of looking at odors for COVID-19. This group is actually in Israel. Hassam Hayek, another researcher in the area, along with some colleagues, he sent some samplers to China in the original outbreak that they had, and they were able to take breath samples and to do some analysis. So there's more than one group around the world that are thinking about this. We have anecdotal evidence that dogs play a key role in detection. And so the trick for us is kind of answering two major questions. What are the chemicals that actually we're detecting? What are the biomarkers that are in the breath? And then how can we have a portable sensor system that can effectively do that detection in many types of settings? So if you want to detect it in people at airports, if you want to look at it on trains, if you want to look at it in big stadiums when people go to sports arenas, that's kind of the two challenges: biomarker ID and then instrumentation. Our team at UC Davis is a combination of a few different types of researchers. We've got some chemists and then me from engineering, but also clinicians. They're a critical part of this. My two clinical collaborators are Nick Kenyon and Mike Schivo. They're in pulmonary and critical care. And I got to tell you, they have worked so hard during the pandemic that it has been so inspiring to be a part of their team and trying to tackle this COVID-19 research problem. So when we think about how to sample exhaled breath, I told you there were two fractions. There's a gas fraction, but then there's a condensate fraction. And the condensate fraction we know has a little under 4,000 chemicals in it. It's about 3,600 biomarkers that you can detect. And so we came up with a way in our group, this is several years ago, this predated the pandemic. We just happened to be ready when it came around. Where we made a MEMS device. So it was a microfabricated device that had features that are very small, so on the order of micron, tens of microns, to condense exhaled breath in a way that was dropwise, not film-wise condensation. And that dropwise condensation would then interact with the surface. It would condense in certain ways, and then those droplets would actually roll across the surface, freeing up a nucleation spot behind it so that additional condensate could be collected, because this was a very energy efficient way to get as much condensation as you can while using a pretty limited surface. And so this entire device, I'll show a picture of it in the next slide or two, but this entire device is pretty small. You can condense the breath and then it collects towards the center point. And again, so it has a free energy driven mechanism where the condensate droplets interact with the surface features and the hydrophobic nature of these surface features. They roll towards the center, and then you collect the condensate in the center part. And so it's a self-cleaning surface. When it's patterned with this spatially distributed wettability gradient, you get droplets and condensate that forms and then it rolls. So again, we're not putting any energy into this. It's a free energy driven mechanism. and then we collect it in the center point. This shows an example of what it looks like from the side. So this is just a syringe that has a droplet of deionized water at the end of it. But what this movie shows is the nature of how it moves. It's actually pretty fast. So you put the droplet down at one edge and it unaided moves towards the center, again, because it sees a wettability gradient. So the leading edge of the droplet, the leading edge going towards the center, is more hydrophilic than the back end of the droplet. And so as the droplet sees its hydrophobic gradient, it sees that it wants to move towards one direction. And so that's why it goes towards that center collection point. This shows an example of what it looks like from the side. This is actually measuring contact angle. One measure of hydrophobic or hydrophilic interaction of droplets with a surface is a measure of the contact angle. And that's this angle here between the edge of the droplet and the surface that it's contacting. And if that contact angle looks like this particular contact angle, then it actually is it's a certain hydrophobic or hydrophilic. As it moves along our gradient, it actually changes its wettability, and that makes it more hydrophilic as it goes towards the center. And so that contact angle decreases as the droplet moves from left to right. Now remember, the pattern is across this entire surface. And we're watching the result of that droplet as it goes from one side to the other. And what is interesting about this also is that you can see that it starts as like a big tire shape or a donut on the right-hand side. And on the left-hand side, you can see it looks like a flat tire. And that's because as it moved, it was very hydrophobic over here, and it's very hydrophilic over here. So now as it goes towards the center, it actually has a continuous change of that contact angle and wettability gradient. But that's how it moves. This is taken with a measurement called a goniometer, and that's how you can measure droplet wettability gradients on microfabricated surfaces. So now this movie shows a little bit about the surface energy that is happening as the condensate is on these different rings. Now, what you probably remembered from looking at this picture a couple slides back is that there's a bullseye of concentric rings, and there's about 5 or 7 of them. And as they move from one ring to another, the pattern on the surface changes, but that's also propelling the droplets towards the center. It's not gravity driven. And so it is a fairly strong interaction between those droplets and the surface, because it's enough to resist other airflow. And so what this shows is, this is a movie taken by a graduate student and a postdoc in my lab. And what they found was that even a compressed gas line, so they used a compressed nitrogen line in our laboratory to try to dislodge these droplets. And I think they were like around 50 to 90 PSI. It was pretty high. And so they were trying to blow these droplets out of the way. And what you can see in this movie is that they resist it pretty hard. So I'll show you guys the movie.
I want to show that exactly they don't want to move.
This is awesome. Just touch the condenser.
Did you see it?
So what you could see from that was that it really took a lot of energy to dislodge those droplets. They have a very strong interaction with the surface, and that's part of what we're taking advantage of. So what we did after we created this device, this surface, which is just a MEMS chip at this point, is we put it into a packaged instrument. So we came up with a way to build a device that's palm size, so it fits in the palm of your hand. It has that chip, the micro-Condenser surface on the inside of it. You breathe through a mouthpiece. So a patient breathes through here. This is just a disposable mouthpiece. There's a one-way valve right here. And so as you inhale, you get air through that one-way valve. When you exhale, it comes back through the mouthpiece, and then it goes through this pathway. This is an aerodynamic filter. as you breathe through here, this twisting and turning of the pathway is actually very cleverly designed by one of my former PhD students to allow this to sort aerosols as you exhale. So the really big ones, the ones that are unusual, we don't like them, they come from spit or saliva, they're big enough that they don't make it through this, this aerodynamic filter. They just end up at this bottom part down here, which is like a spit trap. And then the small ones, because this is an aerodynamic filter, the ones with the mass and size range that will go all the way through, they make it through, they're not contaminated with spit or saliva, and they end up condensing onto that condenser surface that's over here. So it's it collects there. We do have a thermoelectric heat sink that's underneath of it to accelerate the condensation process. And then basically over time, you have condensate that collects at the center ring of that surface. And then we can take that and analyze it for chemical content. So when we wanted to try this out, again, this is a handheld device. We wanted to try this for many different things, but we had an opportunity to look at pediatric patients with asthma. These are teenagers that were aged 13 to 18. We gave them the devices. We told them to go home and take exhaled breath measurements twice a day, once before they ate breakfast and once before they ate dinner every day. And then we got them to take their condensate, and they just were able to pour it into a little vial and then stick it in the freezer at their house. And then at the end of 2 weeks, we came back and we collected that. To be able to analyze it in mass spectrometry, we had to take it through one more step. We lyophilized it and then reconstituted it in a buffer that made it okay to put in a mass spec. It was a solvent, specific solvent. And so this was then fed into a commercial instrument called an LC QTOF. And this instrument does really high resolution mass spectrometry. And then when we got data out of it, it was two types. One was untargeted analysis. We could ask the question, what all was in there? The answer we know now is about 3,600 chemicals, so it's a lot. The other thing we could do is we could target compounds. Now, these teenagers had asthma. We know asthma is an inflammatory condition. And so we asked the question, if we had a panel of inflammatory markers, could we look for those in the exhaled breath condensate? Nobody had done that before. And then could we then do multivariate or univariate analysis on these untargeted or targeted biomarkers to tell if there was a difference between the pediatric patients who had asthma and the subjects who were, for all we know, normal, healthy control. They had no known pulmonary conditions. And what we found was really interesting. A couple of key observations. One is that we are more alike than we are different. So this is out of the patients that were in this, there were about 14 patients in the two categories. And this shows the clustering that you see across all of those people. This is, again, about 4,000 chemicals that are getting measured. And when you compress that data into something called a cluster plot, that is called principal component analysis, it allows you to compare kind of a vector of data and how similar or not similar it is with other people's panel of their chemical abundances. And so this is the dotted line here is the 95% confidence interval. So there's very few outliers. There are a couple over here for this person. This person had some breath samples that were less similar to the others. But when you take out the outliers and which is very few in the overall picture of things, and then you just look at what's left over, there's two key observations. One is that when you look at them by person, so pink star is one person, blue triangle is another person, green square is another person, that you can track a person's metabolite profile and you can tell them apart. So you could tell apart one person from another, but you can also track it over time, and they are very similar to each other. So you don't tend to deviate from your own baseline that much during a 2-week period where you're taking measurements every 12 hours. This was really interesting. Nobody had shown this before. And then at the bottom in the left-hand corner, when you separate these, this is the same set of pictures like here and here, but they're just colored differently. when you make the people who had known clinical asthma, when they're red, and then you see the control people are colored green, what is more interesting is that they differentiate very clearly, that there's a very clear line that differentiates between those two clinical phenomenon based on untargeted metabolites. So in this case, it's all 3,600 chemicals. We don't really care which chemicals are different. We just know that there is a big difference between people who have that condition and who don't. But remember, we went into this thinking that inflammatory markers are probably pretty significant. We wanted to at least test that hypothesis because we know that asthma is an inflammatory disease. That's part of what happens is you have inflammation, people's lungs constrict, and then they wheeze and they're not able to breathe as freely as they did when they have an asthma attack. So we went in and we looked at, again, this is for a panel of inflammatory markers. So rather than looking at all 3,600 chemicals, what if you just look at these? These are the ones on the far right-hand side. And these chemicals are known inflammatory, not necessarily known to be up or down regulated in asthma, although a few of them are, but inflammatory markers in general. And we were able to measure all of these. And when we only looked at this panel, which is a much smaller subset, and then we also looked at what were the differences between asthmatic and healthy control, you see an even more exaggerated difference. So we kind of cleared out some of the noise. We zeroed in on compounds that appear to tell a better indicator of whether somebody had asthma or not. What was even more interesting is when you look at that panel of inflammatory markers, these break down by different biochemical pathways at the cellular level. So the P450 pathway results in some inflammatory markers. That's this one right here. The COX pathway has even different ones. And these are the inflammatory markers that are associated with it, many prostaglandins. The LOX pathway, and then several others are a couple of other different pathways. Each one of those pathways represents a drug target. And so when clinicians give asthma drugs to patients, they usually try one. And if it doesn't function for that person, they go, okay, well, it didn't work. Let's give you a different one. And they give them the next one and they try. It's kind of trial and error. And so what was interesting to us is that we could measure all of these biomarkers from these different pathways, but that they were different in those two groups. Now, what was surprising to us was that the asthmatics were lower because we had anticipated that they would be higher because we know asthma is an inflammatory disease. So we were puzzled by this. And we went back and we looked at the recorded inhaler use and also the systemic drugs that these particular subjects were on. All of them were taking either inhaled or oral corticosteroids. So now this made sense because as many of you probably know, steroids suppress inflammation. And so they were already on medication that was suppressing their inflammatory markers. So they didn't have any symptoms when we were taking measurements from them. They were already on medication that was suppressing the inflammation for their asthma. And we could see that in these pathways. They were diminished in the abundance of that chemical, that potential marker was diminished in the asthma patients because their drugs were diminishing that amount. This is really interesting for another reason. It kind of brings up an idea that if you can predict what kind of response a person might have, you might be able to tailor their therapy a lot better. So rather than just having non-specific inhaler use, that you could actually predict, okay, this is going to be better for this person and this one's going to be better for this other person. So I think this has a potential for individualized medicine that is, we haven't realized it yet, but I think that that's possible that we could realize that in the future. So we had a pilot project this year with CITRIS, kind of a culmination of a lot of things. We know that COVID-19 is an inflammatory disease. That's one of the reasons that people end up, sometimes some people get really sick, is that their inflammatory processes go wild. They just are, they're dysregulated. And so we had a hypothesis that our breath test could potentially not only diagnose COVID-19 infection, but that we could track it and track whether people are getting better whether they're getting sicker, and maybe predict the people that actually are most at risk to needing medical intervention, so you could keep a closer eye on them. To do this, we wanted to compare metabolite profiles from normal healthy people to people who had confirmed COVID-19 infections. And then separately, we wanted to compare this to seasonal influenza patients. It turns out we had already been running a clinical trial to look at seasonal influenza prior to the COVID-19 outbreak. We'd been running a flu trial for about 3 years. And so we had a bank of exhaled breath condensate samples from those influenza patients that when we went into the pandemic, we already had breath samples from them. And then secondly, because our device is so easy for people to take home, we thought, well, what if we send it home with them and we get them to monitor themselves at home as they convalesce after they've gotten sick? And so these are both underway. I'm going to talk about a little bit of what we've been up to. Early on in the pandemic, we did have some trouble collecting breath samples for a couple of reasons. Originally, when the doctors and nurses in the clinics were slammed, there was an overall shortage of personal protective equipment, PPE, and that had to be conserved for clinical use. And so we were not able to take samples in the first wave. We were able to take samples starting in about July and August up to the present. But in the first wave in last spring, we didn't collect any samples. The other thing that happened, and this is something that happens in clinical research a lot, is that you have people on your team that are called clinical research coordinators, and they're doing other stuff. In this case, UC Davis was participating in both the Pfizer and Moderna trial, especially the Pfizer trial. We had a ton of people enrolled in it up at UC Davis. And that meant that those people couldn't take samples, and the clinical research coordinators could not take samples for us as well. So now the vaccine trial is over, and we have 3 clinical coordinators that are dedicating a half day a week to collect our samples. Just in the last couple of weeks, we've gotten more than half a dozen. This is from like a week ago, so it's gone up by now. And we have sample collection occurring at 2 different sites. One is at the UC Davis Inpatient Medical Center. These are for people that have gone into the hospital because they're sick enough that they need to be watched. Some of them are in the ICU, some of them are just in the hospital, but on the regular ward. And then we also have permission to sample at the VA, the Veterans Administration Hospital in Mather. And we're collecting from both of those sites now. So we have a little under 20 samples to date that are from COVID-19 patients. And then we have an equivalent number of samples from normal healthy controls. And those are also tricky because many people are asymptomatic. So you actually need to make sure those people are getting tested and that you know that they're truly not infected. So we have those 2 groups right now. We hope to actually get some asymptomatic breath samples as well, but we have not made that a priority so far. So once we've collected these, as you could imagine, these are infectious. So they actually need to be handled in a BL3 laboratory. That's a special kind of laboratory where you can handle really infectious material. Our collaborator, Sakar, is the chair of medical microbiology at the UC Davis Medical Center. And so we're taking the samples from one part of the med center over to her laboratory. And then she follows our sample preparation procedure. And then at that point, they're deemed non-infectious, and then she can bring them out to our laboratory because at that point, they don't have any virus in them anymore. And so then the last part of that is by the time they're non-infectious, then we can then take those samples and we can run them on our mass spectrometer, which is that LC-QTOF, to look at the metabolites that are present. So just to kind of segue just a second, one of the things that we also look at is how are these biomarkers generated? We've done a lot of work over the years using cell cultures as a way to mimic the breath sample. So they're getting generated by cells in the body. So we do it in vitro as well. And we have a lot of data showing rhinovirus and influenza virus actually changes the metabolite profiles that you can measure in culture in the lab. We've done this with influenza. There's 3 different types of strains of influenza that we've used before. One is H1N1, that's the normal swine flu that you would see, or the human flu that was responsible for the flu cases in 2008. And then avian influenza constructs. These are not pathogenic necessarily in people, but it could be. And so we've infected our cell lines with these, and we can also see those VOC and non-VOC markers. This is a way to predict in a cell culture system. You could predict ahead of time what type of breath biomarkers you would see based on doing it in a cell culture mimic for an in vitro study. So we did this, and we found that you can see multiplicity of infection, which is how much virus load you get in your cells. That directly correlates with the type of biomarkers that come out of our cell cultures and us. And that is also, there are 2 types of biomarkers. There's some non-specific that are just related to all flu. And then there's some specific ones that are related to the type of flu and how much of it you got. And this was something we did a few years ago. So just to kind of wrap up, we would very much like to look at those cell culture biomarkers as a predictive way before the next pandemic happens. Can we be ready? Can we have a cell culture system that could predict the breath biomarkers for emerging pandemics in the future? Because that would shorten the time that it would take to come out with predictive markers for when a new infection is circulating. We actually did collect those seasonal influenza samples over 3 years. We've already analyzed those, and they do a very good job at predicting whether people had the flu or not. But what's also interesting, we took samples from people pre and post flu vaccine for 3 years. And that also, the breath biomarkers change just when you get vaccinated. And so we're also going to try to extend that to COVID-19 vaccinations as that rolls out to the general public. It's a little harder to do that study right now. So that's where we're heading. We have one project that just started that's an NIH funded project to look at the VOC biomarkers of COVID-19 using that gold standard mass spec. We're developing a point of testing diagnostic platform that you can do the whole thing end to end. So you can not only just take a sample, but that you can measure it, that you can get an answer in 5 minutes, and then you actually have a way to tell people whether you have an indication that they're infected or not. We're coupling this with some machine learning to try to go through that data in very short timeframes. And then part of that project is actually to look at commercial plans and regulatory approval. So the NIH specifically wanted a pathway to make sure that there's a product or something viable that comes out the other end. So it's a little more than basic science. So I want to give big kudos to the people in my group. This is obviously pre-pandemic. We don't have any masks on here, but we're definitely doing social distancing and everything now. A lot of really talented people in my group and in prior years. We have tons of collaborators that have made this work possible. And then also we've had some very good support from several funding agencies, including this latest grant from the NIH, which I think is going to be really exciting to work on. So with that, I will stop, and I am happy to answer any questions.
So thank you very much, Cristina. That was fantastic. So there's a comment and a question in the Q&A. The comment is that apparently CNBC has reported that Germany have been able to detect 94%, to have a 94% detection rate of COVID-19 using sniffing dogs. Do you know anything about that, or how does that fit into?
Yeah, that was one of the studies from back at the beginning of the talk. There are a couple of studies with dogs that have shown really high accuracies. Now, the study sizes are not that large, so they're usually under 100 people. But it's pilot data. And those studies have shown that there's definitely something there. The tricky thing with dogs and with canines is, in particular, they get tired over time, and so you can't just work for hours and hours. They need breaks, and obviously, to be rolled out widely without having to train every animal that's participating, it's better to move towards analytical instruments if we can. And yet those dog studies were really fundamental. So I'm quite glad that they did them. I'm also really glad they published them, because a lot of the dog studies for years, they'd show up in the media, but they didn't actually publish them in scientific journals, but they're doing it now, which I think is a good step.
Thank you. And Amy writes another question. So first, she thanks you for excellent and timely talk, but she wants to know that, can you elaborate on potential for innovation? In particular, are there opportunities for simple bridging cell culture systems to quite complex human subjects?
I think so.
Introduction of intermediate or complexity organ on chip?
Yeah, that's another area. I didn't talk about that today, but organoids on chip, any type of like bridging the gap between pure cell culture and pure people. There's kind of two major steps in there that we see. One is looking at organoids, and we are doing some work in that area right now. It's not published yet, but we see that as a stepping stone. And then the second thing are animal studies. This is an area where my group has not done very much, but I have some collaborators that have done mice and rat studies for breath analysis. But more importantly, animals that are really good flu models. Ferrets and mink are two animals that are widely infected by the same types of viral respiratory pathogens that we are susceptible to. And so those animal models are probably a key link in what we're able to do in terms of biomarker discovery, but also validation.
Thank you. Amy also asked the second question is that, do you think, what are the potential and limits to scale this technology? And she wants to know whether reliance on mass spectrometers for detection would be a limiting factor.
So we definitely are using mass spec right now, but that's because it has exquisite resolution. So we would not necessarily recommend mass spec as a scale up type of technology, because it's not available at all medical centers and it's not really widely dispersed in the population. So you would have a disparity between rural health and like urban health centers. So what we see is the next step is to try to translate, now that we know the knowledge from the mass spec, can we then take that to a platform that is scalable and that can roll out to resource limited settings, so that it's not just, you know, gold standard university medical centers. It could actually go out into the communities, and that's where we see it heading.
Thank you. And then David writes that, is there a prospect of pool sampling in a room, for example, with people who have, you know, COVID-19, pneumonia, and so forth? And specifically, it says this could be very helpful in prisons and nursing homes and things like that.
That's a great question. So what is interesting about that is there's kind of two things. So you guys are probably aware that there was some sewage sampling that did this basically in pooled samples under dormitories at Arizona State University, and it had really high accuracy. Conversely, there are a couple of breath studies that look at crowdsourced breath. They happen pre-pandemic, because now there's really not a lot of examples of that, except for maybe nursing homes and prisons. But one of the key studies in this area that I would highlight is there was a study where people were looking at exhaled breath that was showing up in movie theaters. So imagine that you have a bunch of people, hundreds of people in a movie theater, and they are exhaling all the time. They're watching a movie. And there was a really interesting study on stress. And what they did is they actually looked at scary movies, and they would take breath samples from the HVAC system of this movie theater. And then they tried to correlate it with the scary parts. And what they found is that breath biomarkers related to stress actually increased during the scary portions, and that they were able to see that in a crowdsourced way. So I think that your suggestion is a pretty good one. We just aren't there yet, but it's a good way to go.
Great, thank you. And I have a couple of questions. One of them is that, do you think that I understand that the science of what you're doing is different from the existing methods, such as using the nose swab and things like that, because you're not looking at the virus, you're looking at the metabolites. Mm-hmm. But do you think that some of your techniques could be used to enhance the existing methods of detection, such as, you know, the nose swab or the saliva, but they would look at metabolites rather than looking at the virus?
I do. I think that probably we're going to need all of these together. And an example that I could look at is looking at people going into a sports arena. Let's think forward a year or two when things are hopefully much better. We're going to be living with some type of COVID for a while, even if hopefully it's not much, but there are going to be little pockets of outbreaks. And we're probably going to hit a point where there's some type of screening when people go into venues where there's just a lot of people packed into small spaces. So you could imagine people going into a football stadium, everybody gets a breath test. And then if it comes out questionable, you step to the side and then they can do one of the nose swabs, or they can do something that's a little more invasive. And maybe you have to wait 15, 20 minutes for that second one, but you could get a confirmation. So I think with more than one test, you'll up your overall accuracy of screening rapidly. And that's where that's one way we could see this helping.
And then I have a question. It's a design question related to your MEMS device that, so you have to have these micro channels so that you can get the droplets coming to the center. Have you guys evaluated, for example, using porosity rather than that? And with because you can get capillary effects, and with porosity, you can maybe get a similar result, but it's easier to make it like that.
We didn't, because so with this one, we were trying to do features by design, but I think what you're talking about is a really good chance of being successful, because roughness also is really related with hydrophobicity and hydrophilicity. And so I could imagine that there could be some alternative manufacturing that you could think about. It didn't even have to be the same design. It could be a different design that might have exaggerated condensate collection that's a part of that.
So I'd like to thank you very much. I don't see any other questions. You know, this was a very enlightening talk and really timely, and I wish you success. I think that any contributions that would help with the pandemic, with the COVID-19 pandemic, is definitely important and is welcome. And thank you very much for very nice work.
Thank you. We're very happy to be here today.